Well, morning, everyone. It's definitely getting to be fall outside, isn't it? I looked at the calendar a little bit, starting to think about Thanksgiving and such, right? The holidays are coming up relatively soon. Next week is going to be awesome in this class. Next week is when I'm going to talk with you about the central dogma of molecular biology. And that's going to boil down to how genes get turned on and turned off. So, get here early, get a good seat, strap in. It's going to be a party. My favorite 2 lectures of the whole year. And then after that, Tuesday, November 22nd, right? It's the Tuesday before Thanksgiving. I'm just kind of curious. Can I have a show of hands of the people who think that they'll be here with me on that morning when I give that lecture on the Tuesday before Thanksgiving? So it's about a third of the people in the room. It'll be a nice, intimate experience, all right? What I would suggest is you all just come down to the front. I'll share my turkey recipes with you and things like that. We'll have a good time. But Tuesday, November 22nd is also a date that's mentioned on the syllabus for another reason. Let me just toss it out to you. If you missed either the first exam or the second exam, and I told you that you need to take the makeup exam, that's when the makeup exam is. It's going to be between 12 and 2 o'clock on Tuesday, November 22nd. So keep that in mind. You get one shot at taking the makeup exam. Don't miss it if you've missed the first exam or the second exam. If you took the first exam and the second exam, just forget about everything I just said. You don't need to worry about a makeup exam. All right? But, with that, looking ahead a little bit, let's get to work now and talk about the business at hand. Here's where I'd say we've come. In the last 2 lectures, and there was an exam that interrupted the last 2 lectures, came in between them. In the last 2 lectures, I talked with you about Gregor Mendel and essentially Mendelian genetics. I should tell you, Mendelian genetics, that's what we've been talking about the last 2 times that I've lectured. It's also known as classical genetics, right, or classical Mendelian genetics. So if people, if you hear the phrase classical genetics or Mendelian genetics, you should know those are roughly equivalent terms. But so I've been talking with you about classical Mendelian genetics. And in the last lecture, I took it a step further, and we got to talking about Thomas Hunt Morgan. I mentioned his student Sturtevant, and their work that pointed to chromosomes being the unit of heredity. There was the information that gets passed from one generation to the next is hanging out on chromosomes, and Thomas Hunt Morgan figured that out. And he further figured out that you could figure out the order of genes on a chromosome through recombination mapping. Okay, so that's still the way that people find out where genes are on chromosomes today in all organisms. It tends to be through the kind of recombination mapping that you heard that Thomas Hunt Morgan was doing. And that gets us up to the early 1900s, like 1910. That was the state of the art. So about 100 years ago, that's what people understood. And but in the next 100 years, lots of stuff has happened, and that's what I want to talk with you about today, to lay the table, to set the table perhaps, to then talk with you about how we use all these insights, because we know DNA is where it is that information is stored, right? We appreciate that now, but that was not really in the textbooks. Nobody was talking about that at the time of Thomas Hunt Morgan. So I want to take us today from saying that heritable information is associated with chromosomes to the point where we can start talking seriously about here's how information is spelled out in DNA molecules. So there's going to be a little bit of a history lesson associated with what happens here. Don't get too caught up with the names of the people. This is partly to some of what I'll be telling you about today, there's a human interest story associated with it. It's kind of interesting. I think it's important for you to appreciate that real people like you and like me are the ones who had to struggle and fight their way through and figure out how this was all working. don't worry too much about the names. Don't worry too much about the dates. Try to keep a broad view of things. Keep track of just the sequence and how it is that these things kind of got unraveled and figured out, because I think if you do that, it's going to help you have a better appreciation for how it is that we not only know what we know, but actually help you to wrap your heads around some of the important features of what it is that I'll be talking with you about with respect to DNA. So, Mendel gives us this idea of particulate inheritance. He knows there's some discrete unit, a chunk, a particle that is carrying genetic information from one generation to the next. He further knows that there's 2 copies of all of those instructions inside of each organism, right? He has that stuff pinned down. Thomas Hunt Morgan figures out that the chunks, the particles, are associated somehow with chromosomes. But he still doesn't know that it's DNA. It wasn't until the 50s that we knew that DNA is where the instructions were spelled out. And you guys, where you're sitting today, are thinking, what's a chromosome aside from DNA? That might be a good question for you to ask, because it turns out that I've not been doing justice to chromosomes so far. I've given you the idea, yeah, it's a big DNA molecule. It is. Chromosomes are big DNA molecules, but they also have a lot of proteins associated with them. In fact, the mass of a chromosome, if you weighed a chromosome, it's about twice as much protein contributing to the mass as DNA. So there's a lot of proteins associated with chromosomes. And back in the day of Thomas Hunt Morgan, separation in a laboratory wasn't done as easily and as well as it is today. And when they would try to isolate chromosomes, they would get not just a bunch of proteins, they'd get some DNA. They knew there was DNA associated with chromosomes, but the separations weren't so great, and they'd also get a lot of carbohydrates, a lot of oligosaccharides, chains of sugars that were also present within the nucleus, and to some extent associated with chromosomes. And so at the time of Thomas Hunt Morgan, early 1900s, if you asked somebody, what is it that's holding on those genetic instructions, once you got past chromosomes, but what is it about the chromosomes, there were quite a bit of debate in terms of what the molecule itself was that was containing those instructions. A lot of people thought it was sugars, oligosaccharides, and those who didn't think it was sugars pretty much settled on the idea that it was proteins. And hardly anybody thought that it could possibly be DNA, because DNA is just too simple a molecule in some sense. It's only got 4 different letters, G, A, T, and C, to spell stuff out with. That's just not enough molecular complexity to be able to store and maintain the information you need to be something as complicated as you are. Proteins have 20 letters, 20 different amino acids. So maybe that makes sense. And for sugars, oh my gosh, it's just, the number of different sugars you guys have seen. We've talked about monosaccharides. There's so many different ways that you can put together a carbohydrate. That looked like it could spell out instructions as well. And so Thomas Hunt Morgan says, look, it's chromosomes. And then how do we move the needle a little bit so that we can then start talking about its DNA? Well, the next shoe to fall, the next piece of information came from a guy named Frederick Griffith. All right, let me tell you, just for human interest purposes, a little bit about Frederick Griffith. He's a British Army physician. And during World War I, as a British Army physician, one of the things that he had to worry about, aside from bullet wounds and things like that, is a lot of British soldiers were getting sick, and many were dying from pneumonia. So he spent a fair bit of time trying to figure out what was up with pneumonia. He figured out that pneumonia was caused by a bacteria, specifically Streptococcus pneumoniae. That's the name of the bacteria, the genus and the species, that was killing a lot of British soldiers during World War I. And he further figured out that there were he figured out ultimately there were 4 different kinds of Streptococcus pneumoniae, but there were 2 that really got his attention, type 1 and type 2, better, more descriptively, one kind of Streptococcus pneumoniae that gave rise to smooth colonies when he'd grow them up on Petri dishes, and another that gave rise to rough colonies. And just literally looking at the Petri dishes. One, boy, that's a colony that looks smooth. Hey, that one's all bumpy and rough, right? So he sees there's two different kinds. And he further figures out that the British soldiers who are dying from Streptococcus pneumoniae infections tended to be the ones that had the smooth bacteria in their lungs as opposed to the rough bacteria. World War I ends. Frederick Griffith stays on as a British Army physician, continues to do some experiments. It wasn't until 1928 that so 10 years after the war, he's still trying to figure out what's up with pneumonia. He's still intrigued. It's like a 10-year process that he gets to this point. And he does an interesting experiment. Along the way, he figures out that he can use heat to kill the Streptococcus pneumoniae bacteria, right? So if he boils these bacteria and then infects a mouse with the boiled bacteria, the mouse doesn't get sick. Right? So he figures out that heat sterilizes, right? No big revelation to you and I, but back then that was kind of an interesting thing to have observed. He finds out that he can heat kill these bacteria, and then he does something kind of strange. He takes heat-killed smooth bacteria, okay? So dead smooth bacteria, and he mixes them with live rough bacteria, and he squirts them into mice. He takes that mixture and he puts it into mice, and he looks to see what happens to these mice. He knows the rough bacteria will get the mice sick, but they won't die. They'll get over it, right? And he knows that the smooth bacteria are dead, and they can't infect the mice. And yet, so what is it that caused him to do this particular experiment? It's a mystery. I can't figure it out. It seems like a pretty strange leap to have tried to even do this experiment, because he should have anticipated what the result would be and been very confident about what the result would be. These mice that got infected with dead smooth bacteria and live rough bacteria should have gotten sick and then gotten better. What happened, though, wasn't what he had expected. Those mice that got that mixture died. They died from pneumonia, and when he squeezed the juices out of their lungs and he spread that out on a Petri dish to see what kind of pneumonia bacteria grew on the Petri dish, he found only smooth bacteria. And he has to do a quick rethink here. What's going on? Wait a minute. Did I put in the dead smooth bacteria in the live rough bacteria, or did I get it mixed up somehow? I better do this again. And he finds, though, that every time he puts in live rough bacteria and dead smooth bacteria, the mice get sick and die, and what he gets out of the mice are smooth bacteria. What's up with that? That doesn't make any sense. That shouldn't be what happened. What's up with it, what he ends up concluding, is that there's some heritable piece of information that is present in the dead smooth bacteria that somehow makes its way into the live rough bacteria and lets the live rough bacteria become different, lets them become capable of being smooth. And then that, in turn, gets faithfully passed on from one round of cell division to the next. It's heritable information. So Frederick Griffith, for the first time, starts to get this idea that you can, and this is the word he used, and we still use it today, you can transform bacteria. And that means, in this sense, specifically, you can take genetic information from one bacteria and use it to get a different kind of another bacteria. And a clue was that he could do this with heat-killed smooth bacteria. Whatever this heritable thing was, whatever that particle that was getting used for that transformation process, that thing must have been heat stable. When you look at what's inside of a chromosome, you see protein, you see DNA, and maybe oligosaccharides, Frederick Griffith knew, and it was generally accepted at the time, that proteins were not heat stable. So you can take them off the list if you heat up a protein. Some of you experienced this morning, I'm sure. You fried up an egg, right? Egg whites are a bunch of protein. You put it on a hot skillet, it gets hot, right? Those proteins get changed. You can't get them back to the way they were before you cooked them. Heat destroys proteins, and yet this heritable thing was heat stable. It could handle being exposed to heat, and we know now that it's not protein. It's a pretty good clue that the heritable thing that's saving that information must be DNA. So in 1928, we get a clue that the heritable information, it's not just chromosomes anymore. We can move the ball a little bit further through these bacterial transformations, and we start to now see some pretty good indications that the heritable information must be in DNA. So here are some pictures that tell that same story I just told you about Frederick Griffith's experiment. I'll tell you, they've cleaned up this version of the graphic in your textbook. The older version is one that I prefer because they would show you, because right now, that mouse, right? What do you think? Is it dead or is it asleep? It's hard to tell, right? You know, from the older versions of your textbook, in addition to having the little curled up mouse there that looked like it might be sleeping, it was clear it wasn't sleeping because it had the universal dead symbol on it. It had a big red X on the eyeballs. All right? So, but the X's aren't there on this version. I need you to put those in your own mind's eye. But what you got here in this one graphic now is the story that I just told you that Frederick Griffith did. And again, there's a human component to this. I really can't figure out what would inspire Frederick Griffith to bother mixing heat-killed smooth bacteria and live rough bacteria. Why would you bother to do that? I just can't imagine why he would have thought to have done that, but he did it, and after having done it, he got a result that was very unexpected, and I think there's a good lesson to be learned there. This is something that happens so often in science. It's when you get an unexpected result, that's where the most interesting stuff is. If you get a result that's not consistent with your expectations, there's something going on that you didn't know about, and that might be something really important. And in this instance, that something was a clue that DNA is where those genetic instructions were being stored. So again, just very quickly, if you take live rough cells, and this is what the rough ones look like apparently, and you put them into a healthy mouse, the mouse gets sick but gets over it, and they're fine. If you take heat-killed smooth cells, S for smooth, the mice don't get sick at all, and they're just fine. But if you mix the two, a live, rough cell, there it is, and a heat-killed smooth cell, the mouse would get sick just as if you had infected it with live, smooth bacteria. How about that? And again, because the heat was being used to kill these bacteria, that was a clue of something that suggested, gee, proteins probably aren't where the action is here. Whatever's causing inheritance isn't likely to be proteins. Maybe it's DNA. Maybe it's oligosaccharides. And that sets the stage then for Alfred Hershey and Martha Chase, the Hershey and Chase experiment in 1944. Actually, they did this experiment and they pinned down that DNA was the molecule 3 years after Frederick Griffith died. Frederick Griffith died during an air raid in London as an army physician in World War II, right? So it's kind of an ironic sort of full circle kind of thing. He starts his work in World War I. He ends up dying in World War II. 3 years after he dies, in 1944, Hershey and Chase pin down that really it is DNA that's acting as the genetic material. And the way they do that is by using some viruses. So get away from the mice. And they used some viruses to do a pretty elegant experiment. Let me tell you a little bit about the virus that they worked with. The virus they worked with is called bacteriophage T2. Bacteriophage is fancy genetics talk for a virus, specifically a bacterial virus. Okay? So they used bacteriophage T2. Bacteriophage T2 is a pretty simple thing. It's got one thing that it does. It's a one-trick pony. It infects E. coli cells, and it makes more copies of itself, and then busts open the E. coli cell so that it can infect more E. coli cells. You guys know E. coli. How many times have we talked about E. coli now? It's sort of the lab rat of the microbiology world. It lives peacefully, happily inside of our intestines. Sometimes you get a rogue E. coli that gives you an infection and gets you sick, but typically E. coli is a pretty mild-mannered bacteria. We live happily alongside of it, and it gets beat up by these bacteriophages, T2 in particular. T2 is simple. Again, it's a one-trick pony. It can only do one thing. It can make copies of itself and infect other E. coli cells to make more copies of itself. It's got a pretty simple life that it goes through. And T2 is also simple in terms of what its composition is, what it's made out of. It's got just two different kinds of molecules. It's got a protein coat, a shell, that keeps its DNA safe, and it's got some DNA. That's it, protein and DNA. No oligosaccharides, no lipids, protein, DNA, that's it. And Hershey and Chase know what's up with Frederick Griffith's work. They know that there are people saying that it still is oligosaccharides. They know that viruses don't have oligosaccharides. And they say, we're going to pin this down and we're going to show DNA is where it's all at. So what they do is pretty straightforward, pretty simple. They used radioactive isotopes to label the proteins of T2 viruses and another radioactive label to label the DNA of these T2 viruses. Two different radioactive isotopes. Remember that from like the second time that we've met for this class? One of those radioactive isotopes is a radioactive isotope of sulfur. That's the one that they used to label the protein. They used an isotope of sulfur, S35. So it's got a couple extra neutrons in the sulfur nucleus for S35. It's radioactive. You guys remember cysteine from the amino acid. Cysteine is the amino acid that has sulfur groups associated with it. A thiol group makes disulfide bonds between two cysteines. And so if you make these viruses out of radioactive sulfur, what's going to happen is the proteins are going to get some radioactivity associated with them. There is no sulfur in the structure of DNA. It's only going to be labeling the proteins. And they used a radioactive isotope of phosphorus, P32, to see where the DNA was going. Because you guys remember already, I'm sure, the phosphodiester backbone, there's phosphorus there. So they were able to label the proteins and label the DNA separately of these viruses. And you got to admit, they're pretty cool structures, right? This is an actual microscopic picture, an electron micrograph. Got your protein coat with its lunar lander modules, proteins that latch on to the surface of the bacteria. Here's the bacteria, and then you squirt your DNA in to do the infection. So they take this setup, and they do this experiment. So they have radioactively labeled protein and no radioactivity for the DNA in this experiment. Here they have radioactively labeled DNA and no radioactivity in the protein. They take two different batches, one where the proteins have been labeled, one where the DNA has been labeled. They let them infect some cells, and then guess what they do? They take these infected cells and they put them into a blender. No joke, they put them into a blender, knock off the stuff that's not part, knock off the stuff that hasn't gotten into the cell, and look to see where do you find the radioactivity after this infection has been allowed to occur. And you guys know what they're going to find out. Here's the blender, all right? you guys know what they're going to find out. The proteins from the coat stay outside the cell. That's not part of the infection. They just go off into the supernatant, whereas the DNA, with its radioactive label in this second picture, that's what gets put into the cell. And what they can conclude, very simply, is that DNA is the stuff that's containing the instructions that allows these cells to make new viral particles. And further, those new viral particles have radioactive DNA associated with them. The DNA is getting passed from one generation to the next of these viruses. And so from this fairly simple experiment in 1944, the point is finally made. The issue is settled. DNA is where genetic instructions are stored. All right? Cool. But now it gets people thinking, how can that be? How could you store information in DNA? People appreciated DNA is made out of just 4 different nucleotides. How could that work as a genetic information? How could you spell out instructions if you only got 4 letters to work with? And how does it even come together in some way that you could get this to work? And so in 44, World War II, people are trying to figure out, how is it that DNA pulls this off? We know it's not sugars. We know it's not proteins. We know it's the DNA. How do you get this to work? It was about 8 more years before Watson and Crick solved that puzzle. That's the foundation. One more clue came along the way from a guy named Chargaff. He, inspired by what he saw with Hershey and Chase, wants to figure out more about what's up with DNA. And he gets a clue by characterizing, by analyzing DNA molecules. He discovers something kind of curious. He finds that for every A in a DNA molecule, there's a T. For every adenine, there's a thymine. And further, for every guanine, there's a cyanine. The molar ratio of A's and T's are the same in any organism that he looks at. He studied sea urchins, but he found that every time, whatever he got is the amount of A, he got the same molar amount of T, and whatever he got for the C's, he got for the G's. Okay? So that became known in the late 40s as Chargaff's rule. For every A, there's a T. For every G, there's a C. So there's another clue for you in terms of how is it that DNA can store this information. And the other clue, so there's Chargaff's rule. The other clue that Watson and Crick had to work with, so here's Chargaff's rule for you as it's spelled out. We got actual numbers to bear this out. Check it out, right? E. coli's genome, all of its genetic instructions, 24%, 25% adenine, 23, 24% thymine. For every A, there's a T. For every G, there's a C. Check out wheat, right? More A's and T's than G's and C's, but for every G, a C. For every a T. Here are Chargaff's sea urchins. Lots of A's, turns out corresponds to lots of T's, and not too many G's corresponds to not too many C's, right? So it's Chargaff's rule spelled out for you here. It got to the point, and here's an exam question for you. I've asked this on exams before. If you look at the old tests, you'll see this on final exams. I'll tell you the fraction of nucleotides within an organism that are A's, for instance, and I'll ask you to tell me the fraction of nucleotides that are G's. See how you would do that? This has to add up to 1. There are only 4 different nucleotides. If I tell you that 33% of them are A, that means 33% of them are T, right? 33 plus 33, 66. That means that whatever's left over has to be split evenly between G and C, 17 more or less for each. Okay? So that's Chargaff's rule. something that's going to be an important insight. You guys know this is how DNA gets strung together, but you know the first people to figure that out are Watson and Crick. Okay, but I've showed this to you now so that you can see these 4 different nitrogenous bases and how they play out together. You got the 3 parts. You might remember from like the 3rd or 4th time that we met, I showed you a DNA molecule. When I was talking with you about polymers, right? The monomer that gets strung together to make a DNA polymer are nucleotides. Nucleotides have 3 parts. They've got a phosphate group, they've got a 5-carbon sugar, and they have a nitrogenous base. There are only 4 different nitrogenous bases. Okay? One of them we call thymine, adenine, cytosine, and guanine. I asked you to remember this back then. Now it's going to become more important, so I'll remind you. We can break nitrogenous bases up into 2 categories. We can talk about purines, and we can talk about pyrimidines. The purines are the A and the G. The pyrimidines are the C and the T. Pyrimidines have 1 ring in the nitrogenous base. Purines have 2 rings for the nitrogenous base. Okay? So Chargaff gives us Chargaff's rule in the late 1940s. Watson and Crick know DNA is where the action is, and they further know that whoever figures out how it is that information gets spelled out in a DNA molecule is guaranteed, absolutely for sure, to get a Nobel Prize. This is a big deal. For goodness sakes, Thomas Hunt Morgan got a Nobel Prize for figuring out that chromosomes is where genetic information is stored. If you figured out, if you could break this code, more or less, guaranteed Nobel Prize. And so Watson and Crick, young guys at the time, barely freshly minted PhDs, had the chutzpah to say, we're going to figure this out. And it became particularly cheeky of them to say that they could figure it out because they knew lots of other people had the same clue. They had Chargaff's rule, they had Hershey and Chase's experiment, including they knew Linus Pauling was interested in figuring out, cracking this code, figuring out how information got stored in DNA molecules. Now, I've already bragged to you. Do you remember about how I told you I had dinner once with Linus Pauling and he put vitamin C all over my salad? And, you know, person I'm happiest to have met. Linus Pauling is trying to figure this out. He already has 2 Nobel Prizes, right? And he's looking for his 3rd. He wants the trifecta. And Watson and Crick, despite the fact they're going up against a heavy hitter like Linus Pauling and a bunch of other folks, still go forward and try to figure this stuff out. And so, what do Watson and Crick have to work with? They know the chemical structures of nucleotides. They know about phosphate groups. They know about 5-carbon sugars. They know about the 4 different kinds of nitrogenous bases. So they know this, they know this, they know this, they know this. They have no idea how they get linked together. They don't know about phosphodiester bonds and backbones. They have no idea about base pairing, but they also have Chargaff's rule, which tells them every time you use a T, you got to fit an A into the molecule somewhere. And every time you use a C, you got to fit a G into the molecule somewhere. And then they got one extra thing that Pauling didn't have available to him and that others didn't have available to them. Just down the hall from them was a woman, uncommon at the time, a woman researcher, a crystallographer named Rosalind Franklin, who was characterizing DNA molecules for a completely different reason, just kind of curious about the structure of DNA molecules. And this is an X-ray diffraction photograph that Rosalind Franklin herself generated from crystals of DNA. Let me tell you a little bit about X-ray crystallography. good chance you've not encountered this before. But X-ray crystallography turns out to be what gives us the cleanest insights, the best ground truth about where within molecules atoms are. And what crystallographers do is they get a lot of a particular molecule they're interested in. Nowadays, typically a protein. And they do all sorts of magic. It's like voodoo to make crystals of these molecules that are composed of just the molecule that they're interested in, and then little teeny tiny crystals. And then they shine X-rays through the crystals, and where the X-rays encounter atoms, the X-rays move a little bit, and on an X-ray film that's on the opposite side of the crystal with the X-ray shining through it, they get these patterns that are called X-ray diffraction patterns. And to you and me, we look at something like this, right? I'm not going to ask you on the final exam to interpret Rosalind Franklin's X-ray crystal diffraction pattern. We look at this, normal people look at something like this, and they say, I have no clue what's going on here. But to X-ray crystallographers, they're able to see things and figure out recurring patterns within these crystals, recurring patterns within these molecules that tell them where atoms are. So the crystal would have sat here, the X-ray would have come in from above, and then what you would see is this scattering of the X-rays. And Rosalind Franklin would see these patterns. There's something here, there's something here, there's something here. You can see it's the same thing in all four of those quadrants. She's able to look at this X-ray crystal diffraction pattern for DNA molecules, and she figures out that within DNA molecules there are some recurring features that happen over and over and over again. She sees one recurring feature that is every 0.34 nanometers. Okay? You guys know what a nanometer is? It's tough to wrap your head around a nanometer. A nanometer is a billionth of a meter, right? Super, super tiny, right? But every 1/3, 0.34 nanometers, there's some feature that happens over and over and over again in DNA molecules. That's the first little blip that you're seeing here. Tells her there's some combination, some pattern of atoms that shows up over and over and over again at 0.34 nanometers. She also figures out that there's another recurring feature that comes up on these DNA molecules every 3.4 nanometers. So 0.34 nanometers, 3.4 nanometers, something else is coming up over and over again. Oh, actually, this is the 3.4s out here. Here, 1 nanometer, there's something that's recurring, and here at 2 nanometers, something's recurring. She figures out from looking at these X-ray crystals that there are these 4 recurring features to DNA molecules. 0.34 nanometers, something happens over and over again. 1 nanometer, something happens a lot. 2 nanometers, something happens a lot. 3.4 nanometers, something happens a lot. And James Watson, kind of sneaky, befriends Rosalind Franklin. Not many other people in the lab apparently like Rosalind Franklin. James Watson pretends to like her, as it's recounted. He gains her confidence, and she lets spill to him those 4 tidbits of information. And the talk is that James Watson has a photographic memory, and so he looked at some of her notes, took a snapshot in his mind's eye, and then scurried back to talk to Crick and said, hey, check it out. I got another clue here about what's going on with these DNA molecules. Leaves Rosalind Franklin completely out of the loop at that point, never even tells her that he's taken that bit of insight away from her. And Watson and Crick huddle together and spend weeks now, knowing that there's a Nobel Prize at the end of the finish line here, trying to figure out how to piece together these 4 different kinds of nucleotides in a way that lets you make a DNA molecule that has these recurring features associated with it. And you know what they ended up doing for weeks was largely playing with tinker toys. They would make models of each of the nucleotides, phosphate group, 5-carbon sugar, nitrogenous base, and they spent weeks with these tinker toy models trying to fit them together in some way that caused them to fit into something that had those 4 different dimensions that Rosalind Franklin had found. It didn't take them too long to figure out that the space between rungs on the ladder, the space between nucleotides on a stack of DNA molecules, that's the 0.34 nanometers, right? That's, we think today about DNA as being like a ladder, rungs on a ladder, each nucleotide stacked on top of the next is one of those rungs. Based on what they knew about the dimensions of nucleotides, if you lay them out flat and you put one on top of the other, 0.34 nanometers gets the job done. So they know they're dealing with stacks of nucleotides. They also know that they have these 2 other dimensions. One of those dimensions is the width between one end of the molecule, one end of the ladder, and the other side of the ladder. And that's 1 nanometer. So it's a ribbon that they're looking at here, and they know it's 1 nanometer across. And what that tells them is they got to figure a way to get a nucleotide, or 2 or 3, to fill that distance from one side of the ladder to the next side of the ladder. A single nucleotide by itself, a little less than half a nanometer. 2 of them next to each other, a nanometer. Hey, now we may be onto something. Maybe what we've got here are stacks of nucleotides, but they're arranged in pairs. The 2 nanometer feature is the distance from here to here on this molecule, right? Because it's a ribbon, but the ribbon is twisted, and so the 2 nanometers, there must be some sort of twist to this ribbon. It's the clue that they get. And the 3.4 nanometers is the distance from one twist to the next twist down the line. So it's a stack of 10 nucleotides. Gives you that full turn of that sort of spiral staircase. So they've got these dimensions. They know their model has to fit. They have the structures, the tinker toys of the nitrogenous bases, and all they gotta do is figure out how to put together the nucleotides so that they get something that looks like this. And literally, they played with these tinker toys for weeks. And what they were doing is they were butting their head against the wall because they thought for every G, when you had a pair, they had to get something that's a nanometer across. They thought that for every G, they got it in their heads, that if you had a G, you had to pair that up with another G, right? And for every A, you had to pair that up with another A. That's how they figured this was all going to come together. But it didn't work. For weeks, they kept trying to smush them together, but one G paired up with another G was too big. It was more than a nanometer, because remember, the Gs are the purines. They've got 2 rings. If you try to stick this in the context of another one, the space between the 2 of them was bigger than what they had to work with. And they figured for every T, you'd have to hook it up with another T, and that wasn't wide enough. What morons, right? After a week, don't you think it would have occurred to them if 2 purines together are too big, and 2 pyrimidines together are too small, the Goldilocks solution here, right? Let's put 1 purine with 1 pyrimidine, and hey, this might actually work. We've got 2 things that are too big to put together. We've got 2 things that are too small to put together. Why don't we take one of the big things and one of the small things and hook them up together? When they finally stumbled upon that idea, it all fit into place. Like in a matter of minutes, They were able to sort the whole thing out. It took them a little while to figure out. They had to refresh their memory about Chargaff's rule, but that gave them the last little clue that they needed. For every G, you'd hook it up with a C. For every A, you'd hook it up with a T. For every purine, a pyrimidine. For every pyrimidine, a purine. And off you go. And you put these twists in it to give the recurring features that Rosalind Franklin had seen, the 1 nanometer, the 2 nanometer, the 3.4 nanometer. And they have a model. This is like a 1/4 size version of the model that they had actually constructed. They have a model that's about 6 feet tall that is the double helix that we now recognize as the sort of stereotypic image of molecular biology. They come up with that model, they look at it, they take a step back, and they say, oh my gosh, we're going to get a Nobel Prize. All right? And what's the next thing they do? We got to write this up. We got to publish this as fast as we can before other people figure this simple structure out. And once we've done that, we're going to pin down a Nobel Prize. So later that day, they wrote a 1-page paper that they published in the journal Science in 1953 that lays out this structure. There's a picture, essentially, that shows what you're seeing up there on the screen. And that gets them a Nobel Prize. How about that? 1-page article. It's a classic scientific research publication. I gave you the citation for it in the lecture notes. If you want, you can read it. It's easy to find. It's a simple paper, and it lays out this structure for which they ultimately get a Nobel Prize. You might be wondering, what happened to poor Linus Pauling? You know, he's a smart guy. He had a whole bunch of people working to help him figure this out. Watson and Crick did it off on their own. Linus Pauling's problem, for whatever reason, he thought that DNA had to be a triple-stranded molecule. He kept trying to figure out how to get 3 nitrogenous bases together to code for information. Watson and Crick never even stopped to think about triple-stranded stuff. They were going with double-stranded, again, probably because they had the benefit of insights that Rosalind Franklin had gotten. All right? A lot of you are probably feeling bad for Rosalind Franklin. You probably should, right? She got gypped. She got cheated. That's pretty clear. Even Watson and Crick acknowledged that in retrospect they didn't treat her very well. Do you think she should get a Nobel Prize? Well, she's entitled, I suppose, but the Nobel Prizes have a rule associated with it. You have to be alive to be a recipient of a Nobel Prize. They don't do posthumous Nobel Prizes. And Rosalind Franklin died of cancer, I believe it was ovarian cancer, before Watson and Crick got their Nobel Prize. So she wouldn't have even gotten it. There's a really good chance, I think it's something we should expect, she got the cancer that killed her because she was hanging out with X-rays all the time, generating X-ray crystal structures. At the time, people didn't appreciate how bad it would be to expose yourself to that many X-rays. And so she, in some sense, gave her life for the insight that ultimately was a key piece of information for Watson and Crick to get them their Nobel Prize. Now, Watson and Crick are still around, right? They're still chugging away. Crick wrote a book, I'll recommend it to you, it's called The Double Helix, that describes everything I've just told you, the story, the competition with Linus Pauling. It's autobiographical, right? He wrote a book, it's called The Double Helix. And how it is they kind of cheated Rosalind Franklin. And then that book got made into a movie about 15, 20 years ago. It's an old movie now, but Jeff Goldblum stars in the movie. So check it out. I think it's on Netflix. You can, say you're watching it to study for the Bio L20 final exam. Okay, but so there's that structure of DNA, sort of a stereotypical kind of image. A space-filling model, not that different from the ball and chain model that I have. Here's what Watson and Crick were bumping their head up against for about a week and a half. They kept trying to pair purines up with purines, one purine with the same purine. Then they also were trying to pair up pyrimidines with pyrimidines. And good grief, how could they have been so dumb for a whole week thinking about this to not figure out if you had a purine with a pyrimidine, the dimension, that 1 nanometer dimension, would get solved. A really neat insight that they saw when they figured out purine with pyrimidine made them very confident they had gotten the right structure, is this idea of base pairing. All right, so purine with a pyrimidine, cool, but it's a little more specific than that. It's not just any old purine with any old pyrimidine. It's G's with C's and A's with T's. That's consistent with Chargaff's rule. When they saw with their models putting a G next to a C didn't do that much compared to what they saw when they hooked up an A with a T. The things on the surface of an A, this nitrogenous-based adenine, had charges associated with them that were complementary to the charges on the opposite nucleotide, the A. So let me just put it to you really plainly, right? Check it out. Adenines can form 2 hydrogen bonds with thymines. This has a little bit of positive charge. You can make a hydrogen bond to the oxygen here with a little bit of negative charge. This has a little bit of negative charge. This can make a hydrogen bond with that hydrogen. That's a really neat fit. If you tried to put a cytosine up there, try to do that. Lift this structure up in your mind's eye, put it there instead of the C. What you're trying to do is you're trying to hook up an amine group with another amine group. That's not going to work, right? Positive charge, but positive charge, negative charge, now we're on to something. Cool, huh? And so just as adenine can form 2 hydrogen bonds with thymine, guanine can form 3 hydrogen bonds with cytosine. They fit. They fit really well, and those hydrogen bonds are going to help them stick together in a nice, neat, stable fashion. And so when Watson and Crick saw that hydrogen bonding potential, in addition to satisfying the dimensions of the molecule, they saw the hydrogen bondings. That's it. We got it. We're done. Let's write this up. Let's collect that Nobel Prize, and we'll be on easy street for the rest of our lives. And that's exactly how it played out for them. So base pairing is an important concept. It's kind of a big deal, right? For every A, there's a T. For every G, there's a C. And ultimately, that's because G's can specifically interact with C's, and C's with G's, and T's with A's, and A's with T's, and G's with A's, G's with T's just doesn't work. This is the way they come together. So Watson and Crick come up with this structure. It's a 2-stranded double helix. They see there's base pairing going on. And another big insight they get from this structure, a big insight you get from the structure, is that there are 2 strands, and the information on one strand is redundant with the information on the other strand, right? If you tell me the sequence in which letters are arranged on one strand, G, T, G, C, right? You don't need to tell me the sequence of letters on the other strand. I can tell you if it was G, T, C, C. Is that what I gave you before? Let's say it is G, T, C, C on one strand. It's C, A, G, G on the other, right? The information on one strand is redundant with the information on the other strand. So there are complementary strands. And that complementarity of those strands ends up giving a very clear insight in terms of how it is that you replicate DNA molecules. For us, it's easy. For Watson and Crick, imagine the revelation. You have one DNA molecule, and you need 2, the cells replicating, the organisms replicating. For Watson and Crick, they're the first ones that realize that all you have to do is separate those 2 strands. okay? And then use the information on each of those two separate strands to make another complementary strand to each of them. That's how DNA must get replicated. How about that? You and I, old news, not a big deal. For Watson and Crick, this was huge. They recognized, oh my gosh, this is how information gets stored, and this is how information gets replicated, duplicated if you prefer, so they can get passed on faithfully from one generation to the next. It all came together. They knew that figuring out how information was stored in DNA was going to answer some important questions, and sure enough, the structure delivered those answers. Now, Watson and Crick, I don't know, I probably haven't painted them in a particularly flattering light here today. If you read the double helix, you'll see that I've probably been unfairly kind to them. They were, by Crick's own estimation, they were slime bags, right? I mean, these guys were sleazy with the way that they were stealing from Rosalind Franklin and making fun of her behind her back while they were still peering over her shoulder to copy her test answers down. And it was bad stuff. But here's something that, since I first read that paper, this classic one-page long paper, I read this, and even as like an undergraduate, when I read this, I thought, that's pretty lame. Let me read to you the last sentence of their one-page paper, right? And believe me, it's on point here. We're talking about DNA replication. The last sentence of their paper, and I'm looking at my lecture notes so I get it exactly right. Check out the last sentence. It has not escaped our notice, and this should be done with a British accent, right? Because it's Crick that's writing it. But it has not escaped our notice that specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That's their last sentence of that paper. The base pairing that they are pointing to suggests a possible copying mechanism for the genetic material. I told you, that struck me as being kind of sleazy, kind of slimy. Why don't you just come right out and say it, for goodness sakes, right? Why don't you just say that if you separate the two strands, each strand has the information you need to make a complementary strand? Why don't you just say that? Why do you say it suggests a possible copying mechanism? You know why they didn't come right out and say it? Crick spells it all out in the book, The Double Helix. He spells it all out. He says, well, here, check this out. We knew the structure was going to get us a Nobel Prize. We knew that. And we had this extra sort of icing on the cake that not only was the structure going to get us a Nobel Prize, but we think we even now, from the structure, can see how it is that DNA gets replicated. So we got the Nobel Prize in the bag, but we also later on want to be able to say, and we figured out how DNA gets replicated, right? But they didn't want to take the chance on being wrong. They knew the structure was right. They weren't entirely sure about the DNA replication part, but they wanted to lay claim to it so that like 2 years later when somebody else was able to demonstrate that that's how DNA replicates, you split the strands and you use the strands, they said, oh yeah, that's what we meant by our last sentence. Whatever the answer turned out to be, they wanted to be able to claim that they had figured it out from the structure. How about that for slimy? To their credit, to Crick's credit at least, he acknowledges that he was being slimy when he put that out. But again, check out the human drama that's going on here with all of this work for 100-some years. Poor Gregor Mendel gets no credit for any of the stuff that he did until he's dead. Right? Humble monk. Thomas Hunt Morgan has these problems with keeping fruit flies in vials during parties. Poor Frederick Griffith dies 3 years before people really pinned down that he was right and that DNA is where the information is stored. And then Watson and Crick are playing these mind games with us in their Nobel Prize winning paper just so they can lay claim to anything that has anything at all to do with the structure of DNA for the rest of eternity. All right? So Watson and Crick publish their paper. They get, huge news. It's front page news in all the newspapers. The genetic material has been figured out. And people pay attention to this news, and they read the paper, and they see this last sentence, and they start thinking, well, Watson and Crick say they think they know how DNA gets replicated. Oh, yeah, sure, I can see. If you separate the two strands, that would work. A pair of guys read this, and they say, well, you know, they didn't do the experiment. Why don't we actually show that that's, in fact, how it is that it works? So a pair of guys, Matt Messelson and Frank Stahl, say this seems to be what Watson and Crick are talking about. You have a double-stranded molecule, strand 1, strand 2, right? Information here is redundant with the information here, right? What you must do is you separate the two strands, and then you use the information here to fill in a new strand, and you use the information here to fill in a new strand, and you end up, you started with one double-stranded molecule, you end up with two double-stranded molecules. For each of the new double-stranded molecules, you've got one of the original strands and a new strand. New strand, original strand. So Matt Messelson and Frank Stahl read the paper, look at this structure, and they say, yeah, that must be how it's done. And they do an experiment to verify this. A highlight of my experience in training sort of to become a biology professor, I spent some time as a postdoctoral researcher at Harvard. I loved the time I spent at Harvard. There were always great talks to go to. But there were some amazing people who were on the faculty, and I'd get to go sit in on their lectures whenever I liked. Postdocs at Harvard are given faculty status, and I could just walk into any class I wanted, anytime I wanted, didn't even have to ask permission. Matt Messelson was on the faculty at Harvard while I was there back in 90 or 91. And I found his syllabus, not online, there was no real internet to speak of then, but I found a copy of his syllabus and I said, hey, this is when he's going to talk about the Messelson and Stahl experiment. Matt Messelson. I could hear him talk about the Messelson and Stahl experiment. So I went and I listened to his lecture, and at the end of that lecture, he goes through the whole Watson and Crick story, similar to what it is I've just laid out to you. He goes through that whole story, and he says, so Frank Stahl and I decided this is what we should do. Matt Messelson says, so this is what we did. He goes through and he tells about this experiment, and his experiment is a simple experiment. What they did is they took DNA molecules. They grew up bacteria with heavy nitrogen, an isotope, not that radioactive, but an isotope of nitrogen, nitrogen-15, that's heavier than normal nitrogen. So they got some bacteria, guess which one? E. coli. They grew up bacteria with heavy nitrogen so that their DNA was denser, was a little heavier than normal bacteria with normal nitrogen. And then they let these bacteria undergo one round of DNA replication, one round of cell division. And they said, what we think is going to happen is that after one round of replication, we're going to start out with dense DNA. And if we let the replication take place with light nitrogen, not the heavy nitrogen, we're going to get dark DNA, the heavy nitrogen, and light DNA, the light nitrogen, mixed together. We're going to have one replicate that looks like this, one replicate that looks like this. The long and the short of it is this. This is dense. This is half as dense. It's the intermediate density, right? And so by growing up these bacteria with nitrogen that was dense and then letting them grow with nitrogen that wasn't as dense, they were able to see the change in the density of the molecules. And they further predicted if you let it go one more round of cell division, you'd have half of the molecules that were these hybrids and half of the molecules that were all light, so they could see these changes in the density from one generation to the next, depending on what kind of nitrogen they were feeding the bacteria. So this is what they predicted. They said that that could be described as a semi-conservative model of DNA replication. And if Watson and Crick's vague idea was incorrect, maybe an alternative would be that you would get a conservative model where the dark strands stay together and you end up with light strands, you can see how this is going to give you a population of DNA molecules that have different density than what you see here. Here you have one population, both the same density, kind of intermediate density. For the conservative model, you'd have one population that was light, one population that was heavy. All right? For a dispersive model, you'd get like this mishmash of heavy and light all sort of scattered together. It would probably be something like this in terms of its density, but here this should be a pretty sharp band when you're looking at it, whereas this would be kind of fuzzy and it would get broken. You wouldn't see this combination here for the next generation. So Matt Messelson and Frank Stahl do this experiment. They grow their bacteria for 2 generations. They look at the density of the DNA in the parent cells, the first round of replication, the second round of replication, and they get experimental proof that says DNA replication is semi-conservative, just as Watson and Crick suggested you might imagine. You peel apart the 2 strands, you use the information on each strand to give rise to a new strand, and off you go. So we talk about DNA replication. Okay. Here's pictures of what they did, right? If you want to look at these later, you can, but they're predicting that they're going to start with just 1 band, then they're going to get 2 bands. They predicted, again, this is what they see for the semi-conservative model. For the dispersive model, they don't see this. For the conservative model, they don't see either this or this. Again, they have experimental proof that DNA replication is semi-conservative. So, Messelson and Stahl solved experimentally the last question that Watson and Crick sort of left hanging. And we're at a point where we can start to talk a little bit about how it is you store information in DNA. That's really what I'm going to do with you on Tuesday and Thursday. So before I get into that, and that's going to be mostly on Tuesday and Thursday, let me say a few last words to you about DNA replication. I've already told you that E. coli's genome is about 4 million nucleotides long. I've told you that E. coli can replicate that genome in about 30 minutes. And I've told you, I tried to impress upon you that that means that whatever's doing the DNA replication is going crazy fast, 2,500 nucleotides a second to be able to replicate a 4 million nucleotide long genome in 30 minutes. The whatever's doing the replicating has a name. A protein does it. What protein? That protein has a name. It's called DNA polymerase. So DNA polymerase is the enzyme that's responsible for doing that business of separating the 2 strands and then using the information on each strand to make a fresh double-stranded DNA molecule. So DNA polymerases are what are doing that replication. Let me point out to you that DNA polymerases are not just crazy fast, they are insanely accurate. E. coli's DNA polymerase goes at a clip of 2,500 nucleotides a second, ridiculously fast, but also insanely accurate. Do you know how often E. coli's DNA polymerase puts the wrong nucleotide in? Right? It sees a G on one strand, and instead of putting a C on the opposite new strand, it puts something else like an A or a T. You know what its mutation rate is? Its misincorporation rate? 1 in a billion. All right? So you actually have to go about 100 rounds of cell division before E. coli even gets a single nucleotide wrong. Right? Ridiculously fast, ridiculously accurate, these DNA polymerases. And we know now stuff about how the DNA is getting replicated. On E. coli's chromosome, big old circular piece of DNA, there's one spot where the polymerases start to replicate that chromosome. Excuse me, they sit down and they go off in both directions from that one spot. Brace yourself for a really clever name. You know what that spot's called? It's on the screen there. It's the origin of replication. That's right, where the replication process starts. So there's one spot on E. coli's chromosome that replication can begin. It's called the origin of replication. And DNA polymerases fire off in both directions from that origin as they do replication. And as they're moving off in both directions, if you look at it, it's not hard to see why it is they call that crotch area there a replication fork. That's where the DNA polymerases are actually hanging out and doing their thing, driving around that circle. So this replication fork gets advanced around in both directions. Eventually the polymerases bump into each other, and you end up with two double-stranded circular molecules when you started with just one. And here's an electron micrograph that shows you in real life what a replication fork looks like. Now, the next slide shows you in a little more detail with a cartoon what's actually happening at one of those replication forks. Here's what's going on. It's a more complicated process than Watson and Crick imagined, and then I have led you to believe. But you got your fork, right? You got a double-stranded DNA molecule here. You got to separate those two strands. And then you got to bring in a DNA polymerase to make a copy of each of those two strands. There are more proteins involved. One of those proteins is called a topoisomerase. It's down the line from the replication fork, and what it's doing is it's relieving with a double helix. If you push that fork forward, what you're going to end up doing is causing all sorts of torsional strain. I should have brought a rubber band with me today, because you can twist up a rubber band, right? But if you move all the twists from one end of the rubber band toward the other end, you get it gets a high density of twists on the one side because you've moved them all. You have to actually relieve that torsional strain. So topoisomerase does that, right? It sits down just beyond the replication fork. You need to have an enzyme that's doing that untwisting, and that's called helicase. Once the strands get separated, you need to have proteins that sit there to prevent the strands from coming back and sticking to each other. Because remember, they have this hydrogen bonding capacity. They like to stick to each other. So you need proteins there to keep that separate. And then you got this guy, primase. Funny thing about DNA polymerases, crazy fast, crazy accurate, also curiously idiosyncratic. You would think that a DNA polymerase would see a single strand of DNA and just get all excited and couldn't wait to get to work on it. That's not the way DNA polymerases work. DNA polymerases won't have anything at all to do with a single-stranded DNA molecule. What they like is to sit down on double-stranded DNA molecules and then extend them. Primase lays down a little bit of RNA, 5, 6, maybe 10 nucleotides of RNA that's complementary to this single-stranded DNA. That, in turn, is a comfortable seat for the DNA polymerase to sit on, and the polymerase then extends that up to the replication fork and keeps pushing then the replication fork, and you go on down. DNA polymerase can't get started unless something lays down that little track for it to begin the business of DNA replication off of. So primase does that. And let me point out to you this difference between a 5' end and a 3' end. We talked about this before. Now is where it really starts to be important. You might want to go back to the first part of the class just to refresh your memory about these structures. But with a DNA molecule on a strand of DNA, on one end you'll have a 5' carbon that's not connected to any other carbons or any other sugars. right? And on the other end, you'll have a 3' carbon that's not connected to anything. That 3' guy is going to have something on its 5' carbon that connects it to another 3' carbon, and that's on down the line. But so we have an orientation to these molecules. There's a 5' end, there's a 3' end. You could think of that as the beginning and as the end, okay? The 2 strands of DNA molecules that Watson and Crick figured out in their model, turns out the way it actually is, the strands are not just complementary to each other, they're also anti-parallel. Deconstruct that a little, right? Parallel, everybody knows that. 2 lines right next to each other. Anti-parallel means that they're not just parallel, but they're parallel in opposite orientations. If one strand is 5' to 3', top to bottom, the other strand is going to be 3' to 5', top to bottom. They're parallel, but they're anti-parallel. Okay? So, zoom out a little bit, and you can see at an origin of replication, you'll have a primase lay down that red RNA primer, and a DNA polymerase will fire off. All DNA replication, all polymerases work 5' to 3'. They only add nucleotides onto the 3' end, never to the 5' end. And so that works well for one of the 2 strands, but for the other strand, you got this problem. You have to keep laying down RNA primers. As the replication fork gets pushed off in this direction, you need to lay down another primer here to allow a polymerase to go 5' to 3' up to the last one. And what ends up happening is you get one strand where DNA synthesis is continuous. The polymerase starts and it never stops till it gets to the end. And you have another strand, the opposite strand, DNA synthesis is discontinuous, and we call that the lagging strand. The leading strand pushes the fork forward. The lagging strand only gets laid down as the fork gets pushed down the line. So that's how DNA replication plays out. Just another look at what we've just been talking about. And I want to leave you with 2 last thoughts. The lecture notes, the outline says something about DNA repair. The long and the short of it is this. DNA is a chemical. It interacts with other chemicals. A lot of times when it interacts with other chemicals, DNA comes out the worse for wear. That's bad. That could give rise to a mutation. Guess what? We've got a lot of proteins that fix those problems. Okay? They encounter damage to the DNA molecule. They use information from the undamaged strand to repair the damage to the damaged strand. Enough said. That's DNA repair. But at the end of lecture on Tuesday, I said, you know what? I have to talk with you yet about one last thing. I'm going to save that for Thursday. Here it is. I thought I'd start with it. I figured it's better to end with it. Remember on Tuesday, I was talking with you about how information in chromosomes can give rise to problems. If you duplicate a chromosome, if there's a gene dosage problem, if you change the orientation, that could cause some issues. Cytogeneticists look at stuff like that. There's one other thing that I didn't quite get to on Tuesday that I want to just mention to you today, and it'll become important again about a week from today. Don't forget, chromosomes aren't just DNA. There's a lot of protein there as well. Okay? The combination of protein and DNA together we call chromatin. Okay? And the picture on the screen now shows you a little bit about some of these proteins. A lot of the proteins are called histones. They fold DNA up into patterns. They help it be more compact as opposed to this long, fragile, double-stranded DNA molecule. And ultimately, they get folded up even further into these fuzzy structures that end up looking like chromosomes with a microscope. Here is something kind of cool, right? It turns out that it's not just the sequence of nucleotides that determines what genes get turned on and turned off. I'm going to talk with you about that a lot on Tuesday and Thursday. But the proteins associated with DNA also can have an important say about whether or not a gene gets expressed, gets made into a protein or not. Here's a neat example. I'm going to talk with you just briefly now about this phenomenon known as imprinting. About 1% of human genes are imprinted. And what that means is this. Remember, you get one copy of your genes from mom, one copy of your genes from dad. You keep them with you for the rest of your life, right? It turns out that mothers package their DNA differently in some places within the genome than fathers do. The proteins get laid down in different ways. And we preserve those differences between our mom's version of the packaging and our dad's version of the packaging for our entire lives. Now that packaging gets reset when we pass that DNA through our sperm or egg producing cells so that we give, for me, a paternal imprint, for some of you, a maternal imprint, right? But we remember that for the rest of our lives. And for 1% of our genes, the difference in the packaging results in a difference in the expression, such that we have 2 copies of the gene for insulin-like growth factor type 2, one from mom, one from dad. The way that it gets packaged in paternal, male transmitted chromosomes causes that gene to be able to be expressed, to be made into a protein. The way it gets packaged in maternal germline cells prevents it from being expressed. How about that? So I got 2 copies of the IGF-2 gene. I'm only tapping into the information in dad's. Mom's not using it. Sorry. Right? So imprinting, the packaging of DNA, can influence the way that DNA, the genes, end up getting expressed. I've given you an example now of how an imprint can result in just the paternal gene being expressed. Don't worry, there are also an equivalent number of examples where the maternal imprint results in the expression and not the paternal. It doesn't, it's not just a guy thing and a female thing. It's pretty equally mixed, but bear in mind that the way that those proteins are interacting with DNA can have an important thing to say about how that information gets tapped into down the line. All right, I'm done. Have a good weekend. On Tuesday, I'm going to talk with you about the genetic code, and we'll really see how it is you spell out genetic instructions. Again, have a good weekend.